Hello everyone, Assalamu alaikum. Welcome to a new PACademia video. I am Mohamed Atef from year 2 and we will be discussing lipid metabolism intended for foundation year biochemistry and year 2 GIT. So what we'll be discussing today, we'll be discussing the digestion, absorption and transport of lipids and various biochemical pathways involving lipids and triglycerides mainly. So starting with a general overview, lipids are really important for a variety of functions. They are used as structural molecules, as phospholipids, for example, in all cells membranes. They are used for energy in beta oxidation, and they are used for the synthesis of a variety of molecules, including certain hormones. Of course, we have cholesterol, which is considered a lipid as well, and it's very important for membrane fluidity, among other functions, including hormones like forming hormones like the sex hormones and androgens. And the main key element in a lot of lipids, particularly fatty lipids, is acetyl-CoA. And acetyl-CoA is used in a variety of functions, which we'll be discussing. Among that, we need to know how these lipids are transported. So these lipids are generally transported through an exogenous or an endogenous pathway. We will be discussing this. And after that, we need to also know what types of lipids there are. So for example, there are fatty lipids or fatty acids, my bad, which are long hydrocarbon chains with a carboxylic acid group at the end. This carboxylic acid group provides some polarity to the molecule. Smaller fatty acids may be transported via albumin in the blood, while larger ones are packed into lipoproteins, which we'll be discussing. There are some fatty acids which cannot be synthesized by the body, and they are called essential fatty acids, and they are required in the diet. Also, some molecules can be turned into fatty acids. So we have ketogenic amino acids, which we can transform into from proteins into fatty acids, then triglycerides, and glucose, my bad, not glycose, can undergo glycolysis, where acetyl-CoA can also be used for fatty acid synthesis. In terms of the transport of fats, we have our main key players, which year twos will know more about. In terms of semester one, you will know more about it in year two. If you are not, if you are in year one, they are called the lipoproteins, and these include chylomicrons, for example, which include triglycerides and cholesterol. And then we have other things like LDL and VLDL and such. So we can generally, once these chylomicrons are formed, okay, we want the fatty acids from these chylomicrons again. What we can do is there is some enzyme found in the endothelial layer of blood vessels called LPL, or lipoprotein lipase, which breaks down the triglycerides into fatty acids, then glycerol 3-phosphate, and forms the triglycerides again, takes them out from the chylomicrons. In terms of the exogenous and endogenous transportation methods, we could say exogenously what happens is dietary fat could be absorbed into the intestines, where it enters the lacteals in the lymphatics, by being packaged into chylomicrons. And in terms of endogenous transport, it will be in the liver via the VLDL to LDL pathways. We also have, as we said, short chain and medium chain fatty acids, which are transported via albumin, while larger ones are packed. The triglycerides undergo a variety of changes depending on the body state, so if we are well fed or if we are hungry. And these include transportation from adipose tissue to the layer, to the liver, my bad, to the blood, and etc. And we will be discussing this. Starting with fatty acid biosynthesis. So we are synthesizing fatty acids, starting by acetyl-CoA. And this occurs in the hepatocyte cytoplasm. It undergoes these reactions as shown. Again, in terms of year twos, we don't really need to memorize the reactions. However, in terms of year one, it depends on your course, of course, but at least in my time, we used to be required to remember a lot of these reactions and pathways and the enzymes and such. So fatty acid biosynthesis makes sense. It happens why? When diets are low in fat or high in carbohydrates or proteins. So we need to compensate by creating some fats, and that's how we create fatty acids in our body. And we require NADPH, which is a molecule that will allow us in creating and undergoing these reduction reactions, which would create the fatty acids. And this NADPH can be created through the pentose phosphate pathway, which we have discussed, which have been discussed earlier in the previous years. We have several important enzymes in fatty acid synthesis, one of them being ACC, which is formed from two biotin containing subunits, so it's dimeric. And this biotin is important because it acts as a cofactor that is required in order for the reactions to occur. So ACC has two forms, one in the cytoplasm of lipogenic tissues, which is ACC1, and one in the mitochondrial membrane, outer mitochondrial membrane of oxidative tissues, called ACC2. And the ACC2 is important because it acts as a regulatory enzyme for beta oxidation, which we'll be discussing later, by inhibiting the carnitine shuttle, which we will also be discussing later. Additionally, the ACC enzymes are regulated by phosphorylation. So phosphorylation inactivates them, and they can be phosphorylated by pKa, which is synthesized due to reactions or due to pathways involved with glucagon and epinephrine. And then we have AMPK, which is found due to high AMP. It also phosphorylates and inhibits ACC. And palmitoyl-CoA accumulation, which will also inhibit ACC. ACC is activated by dephosphorylation, and this dephosphorylation occurs when PP2A polymerizes the ACC enzyme, and PP2A activation occurs due to high insulin levels, and it's deactivated by glucagon, as we mentioned earlier. The rest of the reactions are catalyzed by the Fas complex, which is a homodimer, so it consists of two subunits, and these two subunits have 7 enzymes each, and they can synthesize 2 fatty acids simultaneously. The reactions are shown here. To summarize, in the first 3 reactions, acetyl-CoA is used to form malonyl ACP, which is then converted to acetoacetyl ACP by beta-ketoacyl synthase, or the KS enzyme. Then in the next 3 steps, there are 2 reductions and 1 dehydration reaction. A butyryl group is formed. Butyryl is then transferred to KS, the KS enzyme we mentioned, and the ACPSH group can bind another malonyl group. This will eventually synthesize palmitoyl ACP. In terms of triglyceride biosynthesis, or what we could say lipogenesis, which is a continuation of the previous slides basically, in order to summarize them, for triglyceride synthesis exactly, we discussed fatty acid synthesis at the beginning. Now we are discussing triglyceride synthesis. It consists of 3 simple stages, being acetyl-CoA transportation to the cytosol, because lipogenesis occurs in the cytosol, and then it gets carboxylated and the fatty acid chain gets assembled. So acetyl-CoA is found a lot in the mitochondria due to the citric acid cycle, which has been discussed before probably. And this acetyl-CoA binds to oxaloacetate to form citrate as part of the citric acid cycle. This citrate can then exit the mitochondria and be broken down into acetyl-CoA and oxaloacetate again, because acetyl-CoA itself cannot exit by itself from the mitochondria. In terms of lipogenesis, we have again in the enzyme ACC, which we discussed earlier. Here it catalyzes a two-step reaction with an intermediate. So it forms malonyl-CoA from acetyl-CoA, and it requires CO2, ATP, and biotin. as a cofactor, as we have mentioned as well. This step is a committed step which allows the rest of the reactions to occur and lipogenesis to happen. Then we have the FAS enzyme, the homodimer we also discussed. And this enzyme is transcriptionally regulated. So what happens is in the liver, FAS in the liver is stimulated by insulin and diminished by polyunsaturated fatty acids. So what happens is basically once there is increased insulin in the blood, it can bind to receptors in liver cells, hepatocytes, and this would allow more transcription of FAS. However, when there are when there is an accumulation of polyunsaturated fatty acids in the liver, these can undergo reactions which would suppress this SREBP in the genome, and it will allow for the suppression of FAS. In adipocytes, they are also inhibited by SREBP, which is activated by leptin in response to excess fat storage. And leptin also helps in decreasing food intake and fatty acid synthesis, and it increases energy expenditure. At the end of lipogenesis, palmitic acid is produced through FAS, and these reactions require NADPH, as we mentioned in fatty acid biosynthesis. This NADPH can be obtained by the pentose phosphate pathway or from malate, where malate is broken down to a reaction happens where malate turns into pyruvate via the malic enzyme. Following that, we need to discuss that triglycerides in adipose tissue are stored. However, in the liver, they are not stored. They are packaged and then released into VLDL. So we do not store triglycerides in the liver. They will be stored if there is an imbalance, and that would cause fatty liver disease. Then we need to talk a bit briefly about glyceroneogenesis, which is basically half-baked gluconeogenesis. It's a fast way to produce glycerol 3-phosphate, and there are two key enzymes that play into it. There is pyruvate carboxylase and the phosphoenol pyruvate carboxykinase. And these are important for the continuation of this reaction to form glycerol 3-phosphate, which can then be used for other pathways. Following that, we'll be discussing lipolysis. So it's a breakdown of fatty acids. Particularly, first we will be discussing triglyceride hydrolysis and then beta oxidation of fatty acids. So for triglyceride hydrolysis, it's pretty simple, at least on paper. What happens is, in order for this process to be initiated, we have hormones acting on adipocytes. This would cause CAMP production and then PKA via production via the G protein pathway. PKA would then activate perilipin 1 CGI58, and this protein would activate the lipase catalyzed triglyceride hydrolysis. So it consists of several steps. The first step is the committed step, where triglycerides are broken down by ATGL, which is an enzyme, into diacylglycerol and F1 fatty acid. Following that, diacylglycerol will be broken down into one monoacylglycerol and one fatty acid by the enzyme HSL. And finally, the remaining monoacylglycerol would be broken down to glycerol and one fatty acid by the enzyme MGL. These fatty acids can then be bound to albumin for transport in the blood, and some fatty acid binding proteins in the cytosol for transport inside the cell, and they are utilized for energy in many cells except the brain and RBCs, unless we are in starvation, which is a bad thing. And now we will be talking about beta oxidation. So beta oxidation is a long pathway, not really long, but you have to bear with me on it. So it's basically a stepwise removal of 2 carbon fragments in each step, and each step has 4 reactions. So we have cyclic removal, and we need to remove 2 carbons in each 2 cycles, okay? So each cycle consists of 4 steps, and these 4 steps are here. Okay, we will be discussing them in more in detail in the next slide. So what happens is this generally occurs inside the mitochondria. However, for very long fatty acids, it can occur in specialized organelles known as peroxisomes. What happens is acyl groups are shuttled into the mitochondria by carnitine. We talked about the carnitine shuttle earlier, and now we'll discuss it more. So we need to insert acyl CoA inside the mitochondrial intermembranous space. However, it cannot enter by its own due to regulatory methods. So what happens is we need to bind this acyl groups or acyl CoA to carnitine to form acyl carnitine, which will enter the matrix and then be turned into acyl CoA and carnitine again. And carnitine can exit and bring more acyl CoA inside the cell, inside the mitochondria. My apologies. And when the process happens, all of it ends. We produce a lot of ATP. In fact, it's more effective than glycolysis. It produces more net ATP per fatty acid compared to um, the equivalent of glucose, for example. So the steps of beta oxidation were shown in the last slide, and to summarize them, they are basically 4 steps. We have oxidation, then hydration, then oxidation again, and then cleavage. Activation of the acyl CoA is the only step that requires energy, and it occurs in 2 sub-steps. In the end, we end with each 2 cycles, we end with 2 acetyl-CoAs for even-numbered fatty acids, and 1 acetyl-CoA and 1 propionyl-CoA for odd-numbered fatty acids. These acetyl-CoA can then be used in the Krebs cycle, and then they could go to the electron transport chain, and the NADPH and the NADH and FADH2 can go to from the Krebs cycle, can go to the electron transport chain and then oxidative phosphorylation, and this would produce huge amounts of energy. Propionyl CoA for odd-numbered fatty acids can be converted to succinyl CoA with some steps requiring vitamin B12, and then it can be used for gluconeogenesis. But again, that's a talk for another time, not even for year 2, by the way. Moving on, we have the process of ketogenesis. So excess acetyl-CoA needs to be broken down or converted to ketone bodies because we cannot keep a lot of acetyl-CoA. And this process happens in the mitochondria. These ketone bodies, through the process shown here, include acetoacetate, beta hydroxybutyrate, and acetone. Acetone being an important one we know. And muscles in general use ketone bodies, including acetone mainly, as energy sources in normal circumstances, especially when undergoing strenuous exercise. However, there are organs like the brain and red blood cells, and cells like the red blood cells, which should not use it. They only use it in processes of starvation, and that could cause long-term damage. Again, these are the summary or a comparison between beta oxidation and fatty acid synthesis. They may seem similar, however, but just opposite reactions. However, they are not that similar. We have some differences which are summarized here. Following that, we need to briefly talk about cholesterol synthesis, which mainly happens in the liver. However, it also happens in the intestines, in the adrenal glands, and in the gonads. And cholesterol is really important. It's steroid hormone synthesis in plasma membrane fluidity, bile acids, vitamin D synthesis, a lot of stuff. And the rate limiting step would be the formation of mevalonate from HMG-CoA. Okay, so first of all, we have HMG synthase, which would allow acetoacetyl-CoA to form HMG-CoA. So it will be catalyzed by HMG-CoA synthase, my bad. And then HMG-CoA synthase, after forming HMG-CoA, the HMG-CoA will be turned into mevalonate. And this step would be the rate limiting step, and it's really important. And it's catalyzed by one of the most important enzymes in our body, which is HMG-CoA reductase. And this is a heavily regulated enzyme in order to regulate cholesterol synthesis. It's regulated by transcriptional methods, by AMP, by cholesterol levels itself, and hormones. And then we have drugs like statins, which are discussed in year two, which are important in regulating this in order to decrease cholesterol levels in the blood for people with hypercholesterolemia and such. Again, regulation of beta oxidation and fatty acid metabolism in general it's heavily regulated. So there is long-term regulation which occurs through changes in genetic expression, and then there is short-term regulation which happens through the effects of allosteric effectors affecting enzymes and covalent modifications. So things like AMPK and other molecules which we have discussed earlier. Again, touching briefly on transport, we know that triglycerides are the main part of chylomicrons and VLDL, while cholesterol is the main part of LDL. That will be discussed again more in year two. And in terms of that, LPL or lipoprotein lipase, as we said again, can free triglycerides from chylomicrons, while the liver can reesterify fatty acids to form triglycerides again. It's important to know that insulin stimulates lipoprotein lipase by dephosphorylating it, and fatty acids and triglyceride synthesis also is stimulated by insulin. And insulin inhibits by phosphorylating hormone-sensitive lipase in adipose tissue. Finally, just to state a few clinical applications, obesity is a very big problem affecting our region and the whole world. And it can lead to a variety of problems, of course, affecting fatty acids and lipid metabolism, since it's our lecture's topic. These include non-alcoholic fatty liver disease, which can lead to steatosis and a lot of problems. And this happens when fatty acid uptake by the liver and synthesis is exceeding the oxidation and export from the liver. So fatty acids accumulate in the liver, and the liver starts to accumulate adipose tissue, or not adipose tissue, fat cells in the liver, basically, adipocytes. Or fat accumulation in the liver, my bad. And then we have alcoholic fatty liver disease, which is not really related to obesity, but it's much more severe because alcohol can be eventually turned into fatty acids through a variety of steps. And this can lead to cirrhosis, which is end-stage liver disease, and it's very severe. And finally, in obesity itself, there are several hormonal changes that occur. For example, adiponectin levels decrease, and that would decrease beta oxidation, further accumulating fat. Leptin levels increase, interestingly. However, there is leptin resistance. So leptin usually makes us thinner, basically. It helps in reducing appetite and such. However, it's not working well. Even though it's increased, there is resistance. Also, perilipin, which we discussed earlier, it's increased due to insulin resistance. Well, that's pretty much it. It is a heavy lecture, but again, be sure to memorize the reactions. At least that was our case in foundation year. And in terms of year twos, it's a general overview with some more additions from foundation year you may find nostalgic. I don't certainly find it nostalgic. And with that, here are my references. Thanks a lot for listening, and see you later in another PACademia video.